Lecture 3 will talk about the drug formulation, especially formulation components and stability of solution. So the main topic of the lecture. So take a look at this diagram. So when you say formulation, what does that mean? So you're basically mixing your active ingredient of your drug with something else than active ingredient itself, because you need to stabilize your active drug ingredient for a long time. So until your drug will be injected to a patient, that structure and the functionality of your active ingredient should be sustained or maintained, right? So then what things do you normally add? If it is a water-soluble drug, of course you will need to dissolve your drug molecule into water or some kind of solvent that will stabilize it, right? And then inside of an aqueous environment, your drug should be able to maintain its structure. So normally your drug molecule, the active ingredient is a protein-based molecule, because a lot of injectable drugs are biological molecules. If that is the case, then you may have to maintain the pH of your drug around the neutral, maybe around 7 area. And then you need to provide enough salt. So it shouldn't have too much salt or too little salt, because salt is the main force for ionic interaction, right? So there should be enough salt in there. And what else should be there? Maybe sometimes you need to add some other stabilizer molecules, like if your drug is aggregating under different situation, you need to provide solubilizer or surfactant molecules to stabilize your drug molecule, its structure. Or sometimes you need to provide antimicrobial agents, so nothing else will be growing inside of a drug. That may be necessary, especially your drug will be used for multiple injection, like insulin shots. That is absolutely required. What else is needed? It depends on how stable your drug is. If your drug is unstable when it's exposed to oxygen or light, you need to provide some protection against that. So we'll talk about all those things that are needed for your drug formulation. Okay, so first of all, formulation components are the things that are added to your active ingredient. Okay, that's what we're talking about. And pharmaceutical formulation is the process in which different chemical substances, including your active ingredient, they are mixed and combined together. Okay, so active pharmaceutical ingredient and molecule is pre-formulated with something else. Okay, so all these things are mixed up together. So proof of concept, exposure, toxicology, qualification, development decisions are made. Okay, at the beginning in the developmental stage. Okay, that may be your active ingredient in this case. And then excipients are selected and added. So there's an excipient process. If it's a pill, of course, there's a bulk agent that should be added. A lot of sugar will be added. If it's an injectable drug, something else will be added. So bioperformance should be checked. Long-term stability should be checked. These are all drug development studies that should be done. Robust process, regulatory approval, user market acceptance, all these things should be considered. Okay. So it's not an easy process. There are a lot of drug formulation scientists and they should work together to find the best, formulation condition for your drug. And then once you find out the best formulation condition, FDA will have final say to that, if that will be acceptable or not. Okay. Most sterile drug, parenteral injectable drug formulation contains at least one additive, at least one, that's minimum, but normally more, multiple. So additive, in other words, excipients. Okay. So that's on top of the active ingredient, that is the major molecule that gives you the efficacy, right? So a major concern, majority contains 2 or more additives. Like I said, maybe buffer or some salt or preservatives, all those things are added normally. And then the largest volume taking component is, of course, solvent. Okay, a vehicle that delivers your active ingredient to the part of the body, human body. So that is the largest component in the injectable drug. Okay, if your active ingredient will be water soluble, of course UFI, water for injection, is preferred solvent, right? That's fine. But what if your active ingredient is not water soluble? Okay, then you may have to use something else. Okay, then water miscible organic solvent can be used in place of water. Okay, for drugs that are completely insoluble in water, then you have to use maybe hydrophobic solvent like oil, vegetable oil. That may be the solvent in that case. Okay, so the solvent normally it should be water. Most cases, the most important solvent in the sterile product, UFI, is the quality of that water that's used according to USP standard. And then water miscible co-solvent could be added. Solvents used to stabilize a certain drug in an aqueous solvent and to reduce hydrolysis. Those are co-solvent, like ethyl alcohol, ethylene, used in cardiac glycosides, and polyethylene glycol in barbiturates, alkaloids, and certain antibiotics. And then injections are given under those conditions intramuscularly due to cellular toxicity. If you inject it through IV, you can kill a lot of cells. You don't want that. So you want a slow diffusion injection. That's why you give it through intramuscular way. The maximum amount of such co-solvent used should be consulted with the literature in toxicology, because you don't want to give too much of those. Okay. If your drug molecule is non-aqueous, it's not water soluble at all, then of course you have to use probably oil-based solvent, liquid fat at room temperature. That's what fixed oil is, mostly from vegetables like corns, cottonseed, peanuts, sesame. Okay, those are vegetable oils that can be used. And hormones and vitamins are prepared with such solution, because those are hydrophobic molecules. Okay. Besides solvent, what other things are added? Like I said, maybe surface active agents or complexing agents. Okay, those are kind of interchangeable words. What they do for your drug is they can increase and maintain drug solubility. Okay, because the structure of the drug, if it's not easily water soluble, then by adding these molecules you can make them better soluble to water. So examples are cyclodextrin, okay, or twin 20, those are surface active ingredients, and twin 80. Okay, so those are examples. And surface active agents, those are used to minimize aggregation of your protein molecules. Protein tend to aggregate by random binding among themselves. Okay, if they bind randomly among themselves, they form big kind of aggregation, means big clump, because one molecule binds with another, and you make hundreds or thousands of binding among themselves, and they lose their structure and they lose their function. They basically become garbage. That's what aggregation is. So you don't want that. So you will keep them separate and safe. That's what surface active agents do. And then tonicity adjuster. So tonicity normally means it's salt concentration. Okay, you want to have isotonic, right? So you don't have too much salt or too little salt, hyper or hypotonic solutions. So isotonic is ideal. So the same salt concentration that your cell has, that's probably the very ideal salt concentration. If that's the case, then you will have any problem when you inject your drug into a human body. Okay, but if not, if your drug has a lot more salt or too little salt, then you will probably create some pain in the injection area or tissue irritation. So you probably want to adjust the salt concentration in your drug if possible. And the antioxidants, okay, if your drug is prone to get oxidized, okay, so then you want to add antioxidant to prevent oxidation of your drug. Inert gas, again, the presence of oxygen may oxidize your drug as well. So if that's the case, then you replace oxygen gas with inert gas like nitrogen, okay, in your filling process, drug filling process in your primary container. And then you can add chelating agents. So chelating agents are what? Like EDTA, they bind with metal ions. By doing so, you can stabilize your drug molecule against oxygen or against the enzymes that can chew up your proteins. Buffers are of course important because what? They adjust the pH. They are resistant to the sudden pH change, so they can stabilize the pH of your drug. So those are added. And then chiropractants, chiroprotectants or lyoprotectants. Those are kind of difficult words, but cryoprotectants. Cryo means what? It's freezing. Okay, so if your drug will have to be freeze dried to extend its shelf life. Okay, that means your drug will go through rapid freezing process during the freeze drying cycle. So during freezing, what happens? Sometimes your water that's mixed up with your drug will crystallize, and then it'll harm the structure of the drug. So then your drug will be probably denatured and lose its structure. To prevent that from happening, you can add cryoprotectants in your formulation. Okay, not only your drug will go through rapid freezing. After freezing, it'll go through drying process. It's called lyo. Is a drying process. So freezing and drying together is a kind of harsh condition. So freeze drying cycle will need extra protectants that can help your drug molecule to go through the cycle. So cryo is against the freezing and lyo is against drying. So you add these molecules to stabilize your drug during freeze drying cycle. Okay. And then competitive binders. What it means is, what if your drug molecule is sticking to the primary container, okay, or tubings during the filling process and things like that, to prevent your active ingredient sticking on the vial, primary container, you may add competitive binders. Instead of your drug molecule binding on the primary container, maybe these guys will bind. So these guys will coat up the primary container, so your drug is not going to bind there. So those are the things like silicone and things like that can be added. And then preservatives, like I said, if your drug is formulated and will be used as a multi-injectable dosage, one syringe, filled syringe can be used maybe several times. Then during that several times of usage, injection, something may be contaminated, can contaminate drug and then start growing there. So to prevent that from happening, it's like insulin shot, multiple injectable insulin shot. You add a preservative in your formulation, then that will prevent growth of microbials in your injectable drug, right? And then polymers, suspending agents, emulsifying agents. Again, those are the molecules that can stabilize your drug structure. Okay, it can control drug release time if releasing time should be extended, and it can also maintain the suspension form of your drug, emulsifying drug dosage form. Okay, so all these things are extra requirements. Okay. So then let's talk more detail about the stabilizing agent. Okay. So co-solvent and amphiphilic compounds. Okay. Besides the major solvent like water, you can add some extra. Amphiphilic, what does that mean? Amphiphilic means it has maybe hydrophilic and hydrophobic parts within this molecule together. It's like STS. Those are kind of good examples of amphiphilic. Okay, so these solubilizing agents should have that chemical structure by binding with the drug molecule, which may have strong hydrophobic part, and it can give a hydrophilic part to your drug. Okay, so that's what they do. Complexing agents and surface active agents are the examples. So let's see, ethanol and propylene glycol are commonly used co-solvent because when they're mixed with water, they will stabilize the polar drug molecules, polarity of your